Hello guys, welcome to this YouTube channel. A good day to everybody watching this video. In this video, I'm going to discuss the answer key for the GATE Excel 2020 paper for the biochemistry section. So let's get started. Which of the following hormones initiates a signaling cascade by directly binding to an intracellular receptor? So this is a pretty easy question because the progesterone here is progesterone. Being a steroidal hormone, usually steroidal hormones actually have their mechanism of action by interacting with an intracellular receptor. So the correct answer here is progesterone. So that was a simple question, and for the others, if you see, insulin, gonadotropin, and epinephrine, they don't act by binding to any intracellular receptor. Okay, moving on to the next question. Which of the following bonds is not present in ATP? So here, if you look at the structure of ATP, which is a nucleotide, you will have a phosphoester bond, you will have a phosphoanhydride bond, and you also have an N-glycosidic bond. However, what you don't have is an alpha glycosidic bond. So the correct answer here is alpha glycosidic bond. Moving on to the next question. The reaction involved in the direct conversion of L-phenylalanine to L-tyrosine. So if you look at the structure, if you look at the side chain, because the main amino acid structure remains the same for both. However, if you look at the side chain, L-phenylalanine has a structure like this, as a benzene ring like this, while L-tyrosine has another OH group attached to the para position. So here, if we were to convert L-phenylalanine to tyrosine, it is going to be hydroxylation reaction. So it's a very simple question. If you know the side chain of all the amino acids, this is highly recommended if you take up any biology competitive exams. Anyways, moving on to the next question. The human major histocompatibility complex, that is MHC, is the correct answer here is both polygenic and polymorphic. Polygenic meaning it is encoded by many genes, and polymorphic is when there are many different forms of the alleles. So this is a very basic immunology facts that you would have gone through during your lectures as well as when you're preparing. But usually, from my experience, I see quite a lot of questions based on MHC complexes, especially when it comes to MHC1, MHC2 questions in many GATE exams as well as in other biology competitive exams as well. Anyways, moving on to the next question. Hargobind Khurana and Marshall Lillenberg elucidated the genetic code by using a cell-free protein synthesizing system. It was found that poly-U and poly-C result in the synthesis of poly-L-phenylalanine and poly-L-proline respectively. Based on these observations, which one of the following conclusions is correct? This is a very, very simple question if you know basic molecular biology. And the correct answer here is D, wherein the codon UUU, because translation is done by reading 3 bases at a time on the mRNA. So codon UUU will specify L-phenylalanine and codon CCC will specify L-proline, because poly-U and poly-C results in poly-phenylalanine and poly-L-proline respectively. So this is the right answer. It's a very easy question to score. Moving on to the next question. Binding of an antibody to its cognate antigen does not involve. So the correct answer here is covalent bonds. So definitely it's not covalent because it's a very transient but yet strong enough to produce immunogenicity. So hence the correct answer here is covalent bond. That's not involved. However, you have electrostatic interactions, van der Waals interactions, and even hydrogen bonds. Okay. Moving on to the next question. A globular protein of molecular weight 50 kilodalton exists as a mixture of monomers and dimers in solution. The most appropriate technique for the separation of these two forms of the protein is. So since it involves monomers and dimers, which are going to be of different sizes and molecular weights, the correct answer here is gel filtration chromatography, which separates proteins based on their molecular sizes or molecular weights, because the large ones get eluted first, followed by the smaller ones. So the correct option here is gel filtration chromatography. Moving on to the next question. Choose the correct order of the molecules according to their ability to diffuse across a lipid bilayer. So we all know the basic principle that like dissolves like in like, which means that for a molecule to cross a lipid bilayer, the more hydrophobic it is, the more easier it is for it to cross. So if you look at that way, then the correct option here is option A, which is CO2 followed by water, followed by glucose, and followed by RNA. RNA being having the negative phosphate group is really hard to diffuse across the lipid bilayer. So the correct option here is CO2 followed by water, followed by glucose, and then RNA. Moving on to the next question. When one glucose unit from glycogen gets converted to lactate in the muscle, the net number of ATP molecules produced is, the answer is 3. If you look at the Cori cycle, which I am not discussing in this video just for the purpose of keeping it short and to discuss only the previous year's questions, if you look at the Cori cycle and understand how glycogen metabolism is being circled between the skeletal muscle and the liver, then you will understand that the net number of ATP molecules that is produced when one glucose unit is removed from glycogen is, and when it gets converted to lactate, then the correct answer is 3. So that's a simple question. Moving on to the next question. Considering the 3 pKa's of histidine, are pK1 equal to 1.8, pK2 equal to 9.2, and pKr equal to 6. Its isoelectric point will be. So first of all, histidine is a basic amino acid, which means it's positively charged. So it can exist as +2, which can, and also +1, and also histidine 0, which is net negative charge is 0. That's the electrically neutral state, and then it can also be histidine -1. That's the maximum negative charge that it can have if you look at the structure of histidine. So here you have pK1, here you have pKr, and here you have pK2. So for calculating the isoelectric point, that is basically your Pi for histidine, you need to take the conversion state of +1, 0 to -1, which is pKr plus pK2 divided by 2. If you look at the titration curve, this particular conversion states will help you determine the isoelectric point of histidine, that is the pH at which it is electrically neutral, that is with +0 as charge. So in that case, upon substituting these values, you will get 6 plus 9.2 divided by 2, which is 15.2 divided by 2, and the correct option, the correct answer here is 7.6. So when rounded off to one decimal place, the correct answer here is 7.6. This is a very simple question to answer if you know how isoelectric points are calculated for basic and acidic amino acids. So it is very important to know which pKs to take for calculating the isoelectric points. Moving on to the next question. 1 mole of a native protein upon N-terminal analysis yielded 1 mole each of aspartate and valine. Therefore, the protein in its native state exists as. So since it's an N-terminal analysis and you get two types of amino acid residues, then it's a heterodimer. So it's pretty simple. However, if it had been just one amino acid, it can be difficult to say from this. But if it's two types of amino acids, then it's a heterodimer because there are two different types of amino acids that are yielded upon the N-terminal analysis. Okay. Moving on to the next question. The prosthetic groups or cofactors involved in both 1 electron and 2 electron transfer in the mitochondrial electrotransport chain include. The correct option here is coenzyme Q and FMN. So here it's important to know how the electron transport chain works in the mitochondria and how the electrons are transferred from one electron transport chain to the other. So it's important to know that, and if you take these into consideration, how 1 electron and 2 electron are transferred, then the correct answer here is coenzyme Q and FMN. Again, just for the purpose of keeping this video short, I'm not going too much into the details because it'll prolong my explanation by too many facts. So this is just like a last minute preparation for your GATE examination. So the correct answer here is coenzyme Q and FMN. Moving on to the next question. This is pretty easy. Match the items in group 1 with the most appropriate items in group 2 and choose the correct option. So here the correct option here is B. So let's see the options. So integrin, yes. It's involved in binding of the cells to the endothelium. Yes, it's a receptor that helps in attachment. And microglial cell, it is involved in phagocytosis in the neural tissue. It's like a different version of macrophage in the neural tissue. TLR7, it is involved in single-stranded RNA recognition. And dendritic cell, being an antigen presenting cell, is involved in antigen processing by cross presentation. So this is a pretty simple question to answer if you know basic immunology concepts. Okay, moving on to the next question. The correct combination of glycosidic linkages present in glycogen is, of course, if you look at the structure of glycogen, you have alpha-1,4 linkage and alpha-1,6 linkage with the branchings and how different glucose molecules are connected next to each other. So this is the correct option, that is alpha-1,4 and alpha-1,6 linkage. Okay. However, I would request you all to know the structures of glycogen, cellulose, and other monosaccharides and different polysaccharides, and even starch, basically the amylose and amylopectin. These are regularly tested in many competitive exams. Moving on to the next question. Polypeptides are either biosynthesized on the ribosomes using an mRNA template or chemically synthesized by the Merrifield solid phase method. The correct directions of peptide synthesis are, so here for mRNA template, if the translation is happening through ribosomes, of course the proteins are synthesized from the N-terminal to the C-terminal. And however, if it is solid phase synthesis, it is the other way around, wherein the proteins are synthesized from C-terminal to N-terminal. So the correct option here is option B, wherein the ribosome-based synthesis is from N-terminal to C-terminal, whereas the solid phase-based synthesis is from C-terminal to N-terminal direction. Okay, moving on to the next question. Solution absorbs 20% of the incident light in a cuvette of path length 1 cm. The amount of light transmitted by the same solution in a cuvette of 3 cm path length is what percentage? So first of all, this is a Beer-Lambert's law based question. So A equal to epsilon into CL, which is log of I by I naught. I naught by I, sorry. I naught by I, that is incident light by the transmitted light intensity. So here for a path length of 1 centimeter, you have A equal to log of let's take I naught equal to 100%, and here since 20% is absorbed, 80% is transmitted. So that will be 80 here, I equal to 80. So that will be 100 divided by 80, which is equal to epsilon into C into 1 centimeter. So this is equal to log of log of 10 by 8. Let's just keep it that way, 10 by 8, which is epsilon into C. Now, if I change the path length of the cuvette to 3 centimeter, if path length equal to 3 centimeter, then the new absorbance will be equal to A prime, let's just call it A prime, that will be epsilon into C into 3 centimeter. However, we know that epsilon into C is equal to log 10 by 8 from this equation. So just substitute that. So 3 into log of 10 by 8. If you know the basic logarithmic rules, we know that log of some a power m equal to m into log a. So I can do this way. So that will be log of 10 cube divided by 8 cube. So this is a new I naught prime by I, right? I prime. So if we can write this in terms of percentage, then how would we convert this? So that will be, but the point is here that we need to know the percentage of the transmitted light, right? That is what is asked in the question. That is, what is the amount of light transmitted by the same solution for a path length of 3 centimeter in percentage. So let's shift the numerator and the denominator here. So the percentage of transmitted light will be is equal to I prime by I naught prime into 100. So that will be 8 cube by 10 cube into 100. So that will be 8 cube is 512, 512 by 1000 into 100, which is 51.2%. Therefore, the percentage of the transmitted light is going to be 51.2% if the path length is 3 centimeter. Okay, so the reason why I'm using this log formula is that it just simplifies my calculation. I don't have to take the log here and then calculate it and then find I naught prime by I prime. So that's going to be pretty messy. However, if I do it this way, it's pretty easy to solve. You just avoid taking logarithms, which can make the calculations pretty complex. Anyways, moving on to the next question. So the second pKa of phosphoric acid is 6.8. The ratio of Na2HPO4 to NaH2PO4 required to obtain a buffer of pH 7 is. So this is basic Henderson-Hasselbalch equation for buffer systems. pH is equal to pKa plus ln of salt concentration, which is Na2HPO4 divided by acid concentration, which is NaH2PO4, right? So this is going to be the second pK is already given for this conversion. So we know the pH is also equal to 7. So 7 equal to 6.8 plus ln of log it is log Na2HPO4 divided by NaH2PO4. So if I take this to this side, it will be 0.2 equal to log of salt concentration, which is Na2HPO4 divided by acid concentration, which is NaH2PO4. So that will be, so now we need to get this ratio basically. So that is what is asked in the question. So that will be Na2HPO4 concentration divided by NaH2PO4 concentration will be equal to 10 power 0.2 if you take the anti-logarithm on both sides. So 10 power 0.2 is equal to 1.58. So the correct answer here is the ratio of the concentration of the salt to the acid is 1.58. This is a very simple question if you know basic Henderson-Hasselbalch equation. Anyways, moving on to the next question. A PCR in a 100 microliter reaction volume containing two primers at a concentration of 0.2 micromolar each is set up to amplify 250 base pair DNA fragment. Consider the average molecular weight of one base pair as 660 Dalton. If the primers are fully consumed by the end of the reaction, the amount of the final PCR product formed is what micrograms? Okay, so this is a pretty logical question to answer. So first of all, let's find the number of moles of the primers used. Number of moles of primers used is equal to concentration into volume. So that will be equal to 0.2 micromolar into volume, which is 100 microliter. So that will be equal to 20 picomoles, right? Okay, so if 20 picomoles of the primers are completely consumed, then the number of moles of the amplicon or the DNA fragment that is amplified is going to be also 20 picomoles. So number of moles of the amplicon is also going to be 20 picomoles, right? If you know the basic PCR reaction. So in that case, let's first calculate the molecular weight in microgram for one amplicon. So each amplicon, if you see, it is 250 base pairs. Each amplicon is 250 base pairs. So in that case, they have also given that one base pair is equal to 660 Dalton. And if you know by fact that 1 Dalton is equal to 1.66 into 10 power minus 18 microgram. 1 Dalton is this. So you need to remember this fact. It's quite surprising that they haven't given this. But anyways, usually it is given in the paper. But if not, it's better to remember this. So 1 Dalton is equal to 1.66 into 10 power minus 18 microgram. So for 1 amplicon, what's the weight in microgram? For 1 amplicon, it is the weight is going to be 1.66 into 10 power minus 18, which is into 660 Dalton, because that is for one base pair. So into 660 into 250, because we have totally 250 base pairs for one amplicon. So if you let's just keep it as microgram, because anyways we have to multiply with the number of moles. So now for 20 picomoles, it will be 20 into 10 power minus 12 into 6.022 into 10 power 23, because 1 mole is the Avogadro number, into, for 1 amplicon, this is the microgram. So we have totally these many amplicons, into 1.66 into 10 power minus 18 into 660 into 250. If you calculate all of this, the final answer comes to be 3.3 microgram. It's just normal multiplication. If you do it with the calculator that they provide, then the correct answer here is 3.3 microgram. So here the final molecular weight of the final PCR product is going to be 3.3 microgram. So this is a pretty tricky question, but if you can understand that the complete primers are used, you will be able to know that, okay, this is the amount of the DNA that's going to be formed, product, amplicons that are going to be formed. Anyways, moving on to the next question. An enzyme obeying Michaelis-Menten kinetics show a reaction velocity of 10 micromole per minute when the substrate concentration equals its Km. So the maximal velocity Vmax for this enzyme, this is a very, very simple question if you know Michaelis-Menten's kinetics. So for a Michaelis-Menten kinetics, V equal to Vmax by 2 when the substrate concentration is equal to Km. Okay, so now we know that V is equal to 10 micromole per minute. Therefore, Vmax is equal to 2 into 10 micromole per minute. So this is going to be 20 micromole per minute. Very simple question to solve if you know this particular fact for Michaelis-Menten's kinetics. There's also no inhibitors involved here, so that makes it even more simpler for this question. So it's a very easy short question to score. Moving on to the last question. The enzyme glucose isomerase catalyzes the interconversion of glucose and fructose as shown. The delta G0 for this reaction is 0 kcal per mole. After adding glucose isomerase to a 0.12 molar glucose solution and allowing the reaction to attain equilibrium, the final concentration of fructose in the reaction mixture will be what millimolar? So at equilibrium, delta G equal to delta G0, right? Yes. So that will be equal to RT ln K equilibrium. So if this is going to be 0 because that is already given in the question, then the concentration of glucose basically and concentration of fructose is going to be the same, right? And the starting concentration here is 0.12 molar. So at equilibrium, this will become 60%. This will become 50% of its concentration, so that the concentration of glucose and the fructose are going to be the same. Half is getting converted, so that ln of glucose concentration at equilibrium, and let's call it EQ, and ln of glucose and fructose concentration at equilibrium is going to be the same. This is going to be same. This is going to be ln 1. So that will be giving you an answer of 0. That's why you have 0 here. So in that case, the concentration of glucose at equilibrium would be 60 millimolar. So that will also be the concentration of fructose, because here it's 0.12 molar. So half of that will be 0.06 molar. That will be equal to 60 millimolar. Right? So half the concentration at equilibrium. So that will also be the concentration of fructose at equilibrium. So the correct answer here is 60 millimolar. Okay. So with this, I am completing the discussion for the answer key for GATE Excel 2020 Biochemistry section. I hope you found this video interesting, and I hope you also found this video useful. And I hope you all have an amazing preparation and ace your upcoming GATE and other biology competitive exams. Thank you for watching. Have a nice day. Bye.